Hello, my name is Firas Khatib, and I'm an associate professor at UMass Dartmouth. I'm going to talk about how the protein folding game Foldit has collaborated with experimentalists, most recently to tackle COVID-19. My colleague, Seth Cooper, already introduced Foldit, so I'll just mention it briefly for anybody joining the session now. Foldit is a graphical user interface representation of the Rosetta algorithm, where proteins can be manipulated with the Rosetta energy shown in real time. So below here you'll see Foldit, and on the right we have a Rosetta simulation with, on the left, its distributed computing project, Rosetta at Home. Now the goal with Rosetta and in Foldit is to reach the lowest free energy conformation, which we call the native state of the protein, or the correct protein fold. Now obviously we don't know if or when we reach that native state in Foldit. All that we have to guide us is the Rosetta energy, which is the Foldit score in the game. Seth talked about our Mason-Pfizer monkey virus retroviral protease success, where Foldit players helped solve a protein that had been unsolved for over 10 years. So I was showing these results in class one day when a student raised their hand and they asked, so you sent the experimentalists the Foldit models, and then those crystallographers tried to fit them into their electron density data to see if it would fit, right? Well, why don't you just give that electron density data directly to the Foldit players? Now this put a smile on my face because that had precisely been our next step. We implemented electron density visualization into the Foldit interface, which you can see here, where players can manipulate their protein to fit the electron density cloud. And they can modify the visualizations for that cloud as well. Now initially, we thought that simply providing the players with this electron density map would be sufficient for them to be able to reach the native state. But even when we provided the players with starting models that were close to the native, and therefore very close to the density, players found better scoring models that didn't match the density at all. Now, this was problematic because as soon as one player gets a high score by ignoring the density, other players don't want to waste their time by folding a losing structure. The breakthrough was when we rewarded the players for matching the density by putting that into the Foldit score. Of course, it sounds so obvious now. It turned out this was the crucial element that we had been missing, ensuring that players are incentivized for fitting the experimental data. Suddenly, Foldit players were successfully fitting even ridiculously large densities. Personally, I can't see anything in there. The obvious next step was to give the players an unsolved case, similar to the monkey virus protein example, but this has actually proved to be quite difficult. I tried hanging out at the back of crystallography conferences, holding a "we'll fit your density for free" sign, but all I got were very strange looks. Fortunately, we were able to collaborate with researchers at the University of Michigan who had just solved the protein, and they were interested in seeing if non-traditional methods could solve it. And if so, how those different methods compared to the standard state-of-the-art methods. So we ran an experiment/competition that consisted of expert trained crystallographers, automated methods, in this particular case, Phoenix AutoSolve performed best, 61 undergraduate University of Michigan students in an Introduction to Protein Structure and Function course, the Foldit players, and Rosetta's automated method for molecular replacement, MR Rosetta. Here's some snapshots of Foldit players building their models into the density for this puzzle. You can see they started with a completely unfolded extended chain and then slowly folded their models into the electron density cloud, which I have to say I am always particularly impressed by, since I can never see how anything fits in these electron density maps, but then again, I'm a horrible Foldit player. Here are the results of the experiment, comparing the best model from each of the 5 different groups. So for all these graphs, the lower the bar, the better. And so you can see that the Foldit players, in pink, outperformed the traditional methods overall, particularly with a lower R-free and much lower Molprobity clash score compared to the other methods. Now, I do want to note that, as was also the case with the monkey virus Foldit prediction, the best Foldit model was not the best scoring one in the game. And I will come back to this later. So after we published this, I just sat by the phone waiting for all the experimentalists to call the Foldit hotline, asking for help with solving their structures. Nothing, unfortunately. The protein structure prediction CASP experiment is run every 2 years, where experimentalists who have solved or are close to solving protein structures release just the protein sequence to the worldwide prediction community. Luckily, during the last CASP experiment, there were some cryo-EM targets. So I contacted the CASP organizers after CASP 13 was over, asking them to connect me with any cryo-EM experimentalists. Not only did the experimentalists provide us with cryo-EM data, after CASP obviously, but before the native was publicly released, they also gave us additional targets that hadn't been used for CASP 13. As you can see in their beautiful figure here, this particular protein complex has many different subunits. It became quite clear that without the cryo-EM data, the players were unable to get anywhere near the native. On these plots here, the further you are to the right, the closer you are to the correct solved structure. The lower you are on a plot, the better your Rosetta energy and better the Foldit score. These two puzzles were run during CASP, and you can see that they got nowhere near the native. So native would be somewhere over here, and the closest Foldit models for this one were very far away, and these ones were nowhere near, completely different fold. Whereas after CASP, but still before the native was released, Foldit players were easily able to fit the cryo-EM data to match the native state. You can see that there are many Foldit predictions that adopt the correct fold of the solved structure. Not only that, but the players converged on these correct topologies very quickly. This is one of the non-CASP targets, where you can see how quickly certain players converged to the correct fold. Also keeping in mind that not all Foldit players play a puzzle in the first few days. We have many different puzzles that are up. People have other things to do as well. But after 48 hours, some players had already correctly matched the native topology. So we again ran an experiment/competition between Foldit players, the microscopists who solved the structures, and 4 state-of-the-art automated methods: Phoenix, ARP, Buccaneer, and Rosetta de novo density. When we compared the key geometric parameters, such as Ramachandran outlier and allowed backbone confirmation, as well as the Molprobity clash scores, between all 6 of the different methods, for all 4 of the proteins in the competition, Foldit produced more realistic models. You can see that with each plot, Foldit here highlighted in green, the Foldit predictions have lower values in all of these cases, and in some cases significantly. In addition to this, for all 4 of the puzzles, the best model was the top scoring Foldit prediction for that puzzle. Whereas you will recall, in the molecular replacement monkey virus puzzle and in the crystal structure competition, the best Foldit model was not the top scoring Foldit one. This, to me, is one of the most exciting results. Due to the absence of the phase problem that exists in crystallography, Foldit's score function seems to correctly reflect model quality with cryo-EM models. Therefore, just by using Foldit, only minimal work will be required by microscopists to obtain an accurate, high-quality model. Seth also highlighted in his talk that we were able to experimentally verify Foldit player designs in our Nature paper from last year. You can see on the right here how close the predicted Foldit designs are to the solved structures. Quite hard to distinguish one from the other, actually. The proteins that were solved by crystallography produced high-resolution electron density maps, which we, of course, gave to the Foldit players to see if they could build even better models in the electron density. So now we have our Foldit community designing completely novel proteins that do not exist in nature, and then contributing to the experimental process for solving their own crystal structures. This brings me to the public service announcement portion of the talk, where honestly, if you are stuck, or if you know any experimentalists that could use a different, non-traditional approach to try to tackle their structure prediction problems, please let us know. Email is right there, folditforscience@gmail.com. You can leave out the dots, it'll still work. But these recent design results mean that not only can Foldit players help with protein structure determination, they can also design novel proteins to fight diseases. Diseases such as coronaviruses. We recently launched 2 challenges in Foldit, trying different strategies to combat COVID-19. The first effort is to design an antiviral protein. Everyone is very familiar with this image from the CDC, with all the spike proteins in red. The idea is to design a protein that will bind to this spike protein, which is exactly what we've done in Foldit. We provide players with the actual spike protein in the game, which they cannot change, as we obviously cannot modify that. And then we let players design proteins to bind to it. Now, whereas the backbone and the sequence of the spike protein is fixed, players are able to add or remove amino acids to their designs, as well as mutate them to any sequence that they want, whatever binds to the spike protein in the best way. Our second strategy against COVID-19 is to design an anti-inflammatory. You may be familiar with the term cytokine storm, which happens when the immune system releases more and more cytokines to defend itself, shown here as white dots. But instead, this causes the immune system to go into overdrive and it starts attacking human organs. The approach we've taken with Foldit is to try to block these cytokine signals and the overstimulation of the immune system. So this is a similar problem from a Foldit point of view. We just need to design those green dots to block the white cytokines. So what makes these problems so difficult? Well, designs need a lot of small interactions with the target. So much so that using purely computational methods won't work. We can't enumerate all the different possibilities, right? There's so many different folds that could fit there, but also for every single amino acid, there are 20 different possible amino acids that we could put in there. So this is where human spatial reasoning and pattern recognition can help when the computational complexity is just too high. But the other tricky aspect is that these designs have to fold up by themselves. Now this makes it non-trivial for citizen scientists who are looking for the ideal interactions, but then they also have to make sure that their model will fold by itself as well. We recently selected 99 Foldit designs as antiviral candidates. These ones were selected from an initial pool of over 20,000 different Foldit solutions. And the main thing I'd like to highlight here is the diversity. You don't have to stare at these too hard to notice how varied these 99 topologies are. So, if you are as eager as we are to find out how these binders performed, well, you can check our blog posts and our COVID-19 video updates as the results come back. Fold.it is our website where you can find everything. And the Foldit project is really the epitome of a massively collaborative project between different institutions, such as the Institute for Protein Design, where all the coronavirus work is done. They publish lab reports every month from the lab, and the Center for Game Science at the University of Washington, Northeastern University, the University of Denver, Vanderbilt University, and most importantly, collaborating with citizen scientists around the world. Thank you, and thank you to the Foldit players, without which none of this would exist.